In this class, we'll try to learn about vesicular transport. Vesicular transport is a process that occurs by either fusion of vesicle or formation of vesicle is called as vesicular transport. Fusion of vesicle with a cell membrane occurs in exocytosis. Formation of vesicle from the cell membrane occurs in endocytosis. So this is example for active transport. Special utilities of vesicular transport are macromolecules such as large protein molecules can't be transported by diffusion or active transport processes. They are transferred across the cell membrane mainly by vesicular transport. Amino acids, sugars, waste products of metabolism, cellular secretions, hormones, neurotransmitter and organisms are transported by this mechanism. Role of vesicular transport. In vesicular transport, formation and transport of vesicles are facilitated by some vesicular transport proteins. So these proteins are Clathrins, coating proteins, diamine, and docking proteins. Clathrin. It's a fibrillar protein located in the membrane beneath the receptor protein. There are two types of Clathrins, AP1 Clathrin and AP2 Clathrin. AP1 Clathrin helps in the transportation from Golgi apparatus to lysosome, and AP2 Clathrin help in transportation of endosomes. Coating proteins. There are two types of coating proteins, coating protein 1 and coating protein 2. Coating proteins help in the transportation of vesicles between endoplasmic apparatus and Golgi apparatus. Diamine helps in the formation of vesicles from the cell membrane, especially in Clathrin mediated endocytosis. Docking proteins. Before exocytosis of vesicles, docking of the vesicle occurs. Docking proteins facilitate attachment of vesicle with membrane. Examples for docking proteins are V-snare protein and T-snare proteins. Types of vesicular transport. It can be of three types: endocytosis, exocytosis, or transcytosis. Endocytosis is a process of transport in which substance is taken into the cell by means of vesicle formation. A region of plasma membrane is pinched off from an endocytic vesicle that causes internalization of the substance to the cell. The vesicle may contain fluid or dissolved solute or particulate material. The size of the vesicle varies from 0.1 to 2 microns. Endocytosis occurs by two mechanisms: constitutive and Clathrin mediated. Constitutive endocytosis. Endocytosis by constitutive pathway occurs in almost all cells. It is called constitutive as the process occurs continually and does not require any specific stimulus. The molecule or the substance make contact with cell membrane that invaginates to form endocytic vesicle. It occurs in two major steps, that is formation of the vesicle and separation of the vesicle from the membrane. Okay, so these are the two steps where the endocytosized substance, they form a vesicle, and then the vesicle gets separated from the membrane. Clathrin mediated endocytosis. This occurs at a specific site of a cell membrane where the Clathrin accumulates. Clathrin is a fibrillar protein molecule having the shape of triskelion, three legs radiating from the central hub. The Clathrin is located in the cell membrane beneath the receptor protein. It can occur in the following steps. The step 1 is protein and other macromolecules attached to the receptors on the membrane surface. The receptor for these molecules are normally concentrated in small pits called as coated pits, beneath which are located fibrillar proteins called as Clathrin. Contractile filaments like actin and myosin are also attached to the coated pits. The attachment of macromolecules to the receptor stimulates the invagination of the coated pit, enclosing the molecule and smaller portion of the ECF to form a vesicle. Clathrin molecule forms geometrical array that surrounds the vesicle. The endocytic vesicle detaches from the cell membrane. The fusion of endocytic vesicle with lysosome that empty their acid hydrolysis into the vesicle during the digestion of substance in the vesicle. Clathrin mediated endocytosis internalizes not only the various organisms but also the substances like growth factors and lipoproteins. So this is example mechanism of Clathrin mediated endocytosis, uh, where initially the macromolecules which are to be endocytosized, they are going to attach on the receptor site. Okay, so then the attachment of the molecules with the receptor causes invagination of the coated pit, forming an endocytic vesicle. So then there will be detachment of the vesicle from the membrane to the cell. So this is another diagram showing the receptor mediated endocytosis. So substances are going to attach on the uh, receptors which are called as Clathrins, and then they get invaginated and gets detached from the cell membrane. Endocytosis is of three types: phagocytosis, receptor mediated endocytosis, and pinacocytosis. Phagocytosis is otherwise called as cell eating. It is the process of ingestion of large particles or microorganisms by specialized cells called phagocytes. The phagocytic cells, such as macrophages, engulf bacteria, foreign particles, and tissue debris and digest them. The phagocytic vesicle vary from 1 to 2 microns in diameter. Phagocytosis occurs in three broad steps. The first is attachment of foreign molecule with the cell membrane, then engulfment of the foreign molecule in the form of endocytic vesicle, then killing of the organism or degradation of the vesicular content. The phagocytic cell includes neutrophils, monocytes, macrophages, and other cells of mononuclear phagocyte system. Receptor mediated endocytosis. Receptor mediated endocytosis is an efficient process of endocytosis in which extracellular molecule binds with specific receptor on cell membrane, as we discussed. Special features of these are receptors are concentrated at the depression of the membrane called as coated pits. Once the external molecule attaches the receptor, the coated pit pinches off from the membrane to form endocytic vesicle, causing a rapid internalization of the molecule. Hormones, growth factors, transport proteins like transferrin, toxins, viruses, etc. enter the cell by receptor mediated endocytosis. Pinocytosis is a fluid phase endocytosis or cell drinking by which substances in solution in extracellular fluid are internalized to the cell. It is a continuous process that occurs in most of the cells of the body. The stages in pinacocytosis are same as that of endocytosis, except for the endocytic vesicle contains fluid instead of solid particles. Exocytosis is the process of export from the cell, which is the reverse of endocytosis. By exocytosis, hormones, neurotransmitter, digestive enzymes, and undigested foreign particles are released from the cells. Steps of exocytosis are: before exocytosis, the molecules are synthesized in the endoplasmic reticulum and packaged in the Golgi apparatus to form transport vesicles. The vesicles then move to the cell surface, fuse with the cell membrane, release the content to the ECF, which requires ATP and calcium. Homeostasis is the term refers to the exocytosis of specific hormones and granules. Mechanism occurs by two pathways. One is constitutive exocytosis and or the second one is regulated exocytosis. Constitutive exocytosis is seen in almost all the cells. By this mechanism, some proteins are continuously secreted by the cells. For example, secretion of mucus by goblet cells of the small intestine. Regulated exocytosis or non-constitutive exocytosis is regulatory exocytosis. Macromolecules that are stored in the vesicles fuse with the cell membrane and release their content in response to specific extracellular stimulus. Transcytosis: when vesicular transport is limited within the cell, the process is called transcytosis or cyto, uh, Pemesis. Vesicles are formed within the cell membrane and transported in the cytoplasm. It is a type of intracellular transport in which various macromolecules are transported across the interior of the cell. By receptor mediated uptake, the molecules are captured in vesicles on one side of the cell, transported across the cell by vesicular transport. It is observed in epithelial cells, endothelial cells, neurons, osteoclasts, and M cells of intestine. Examples for vesicular transport of uh, are IgA, insulin, and transferrin, okay, so which occurs within the cell. Transport across the epithelia. Epithelium is a layer of the cells present on the basement membrane. Two special features of epithelium make it permeable to various it makes it permeable to various solutes. First, the basolateral membrane contains active transport system that creates a gradient for transfer of ions from apical membrane and various uniport and antipodes that regulate exchange of other ions. The second is tight junction between the cell allow selective ions to pass through and prevent large molecules like proteins to be transferred. By two mechanism, the transport across the epithelia occurs. One is transcellular transport, another one is paracellular. Transcellular transport is through the cell, that is from the lumen through the apical surface into the cytosol, and from there through the basolateral space or through the basal membrane into the interstitial fluid. Paracellular transport means the transport through tight junctions between the cells by passing the transport through the cells. Reabsorption of the sodium and glucose in the kidney tubule and intestine is the example of transport across the epithelium. So this is the diagram showing the transcellular transport and paracellular transport. Okay. So transcellular, it occurs across the cell, and whereas paracellular transport, it occurs paracellularly. So this is another diagram showing paracellular and transcellular pathway. Transcellular, the transport occurs across the cell, whereas the paracellular, the transport occurs across the tight junctions. Ultrafiltration through the glomerular capillary, endothelium, and Bowman's capsule epithelium is a special transport mechanism through epithelia. However, in ultrafiltration, transport of substances occurs mainly due to the difference in the pressures across the epithelium. So, this is in brief about uh, the transport across cell membrane by vesicular transport. Thank you.